as my brief chat with Jisong, seems like after he started his own lab in Tsinghua, he's already made some great discoveries in his own lab. Let's welcome Jisong back and deliver the last talk of the symposium.
Okay. Thanks for the introduction. It's also my great honor to present the work developed in our own lab. And today I would like to share with you a story which might shine a little light on how the memory traces is formed in the mammalian cortex. So as we all know that human, as the most intelligent creature on Earth, have the biggest brain relative to its body size. And the neocortex is so large that it is believed that it contains a lot of long-term memories. And it is the long-term memory that allow us to sense the environment, to make imagination, to make creation, to do a lot of high functions. However, how this long-term memory traces is formed in the cortex up to now is still unclear. But it is not totally unclear process. The study is mainly by HM, by studying the patient of HM, characterized by researchers in MIT, and also on some non-human patients, on non-human primates. Sorry. Reveal that this process of memory formation model about two-stage memory system. So memory are thought to be stored in the hippocampus very quickly. And then through a very slow process of system consolidation, it's gradually formed in the cortical modules. And this process has been proved in a lot of studies. However, to really review such process and really know how the brain is encoding the memory, need new techniques to trace the formation process of individual neurons. So we need a method to screen large population of cortical neurons. And also, we need this measurement to be in the single cellular resolution. And also, this recording should be very long-term in order to trace the whole process of long-term memory formation. Now, there are two classes of analysis mainly exist. One is so-called online analysis. People developed genetic coded calcium-sensitive dyes and voltage-sensitive dyes to reveal the events of spikes in neurons that responds in milliseconds or tens of milliseconds. And it can reveal simultaneously hundreds to thousands neurons. However, for larger volume of the cortex, this technique is hard to record because now the imaging technique cannot image so very large, big volume of the cortex. And another strategy is so-called offline analysis. So people utilize the advantage of immediate early gene. So the immediate early gene is the class of gene that be activated by the neuronal activation, like spikes. And those activation can reflect in the expression of those genes in minutes to hours. And they have been shown to be very closely related to the behavior, like memory and memory recall. And it's very easy to record in a very large scale. So a lot of a lot of people have take efforts in this process, and the C-Force reporter and ARC reporter have been generated. Today, I would like to talk another reporter called ZIF268. It's also called EGR1. So this reporter, we could see using the in vivo two-photon imaging in the cortical areas. And you can see in a 300 micrometer diameter volume, we could review around 3 to 5,000 neurons simultaneously from layer 1 through layer 5. And if you closely look at them, the expression intensity of the reporter actually distribute variantly from layer 1 through layer 6. And this might represent the activity difference in different layers. And if you look at the brain slice, and you could very easily tell that different cortical areas have the different activity according to that. And those signals are actually driven by neural activity. And if we measure the expression of EGFP signal, the reporter signal, sensitized rate, according to the dynamics, we can get a sense of about 1 hour delay for the expression of one stimuli-induced signal. And this signal seems to be very sharp, so that it have a relatively high temporal resolution. So with this method, we could measure the living mice for 3 months. And in the first year, when we developed this method, my students quantify the GFP signal for each individual cell by hand. However, this process is too painful, and a lot of them give up immediately. Because each volume, we need to quantify 3,000. And each condition, each mice, we will quantify about 6 volumes. And each condition we will consist about 24 times measuring, so it's a huge number of quantification. So we spent another year to develop automatic segregation system to using the computer to segregate those, identify those signals, so that we can detect the neurons according to its shape, but not to its intensity. Then this segregated signals can be plotted in the digitized manner to study, to trace the changes caused by particular memory formation. And this method obviously has some pros and cons. Compared to other immediate early gene mediate methods, this is much brighter signals, allow the tracing of almost 80% chemokines 2 positive neurons within a volume. And it's also showing very great activity-dependent expression. And EGR1 is already being shown to be related to the maintenance of long-term synapse potentiation and also the long-term memory. However, it has very high background. So even in the naive states, a lot of cells show GFP. So you cannot tell whether a neuron is activated during the task, just according to its brightness. Also, its expression may have some effect of cell type specificity. So in order to balance that, we need to do a cell-by-cell-based calibration before and after the training, and get the whole idea how this signal dynamics can reflect the neural activity within the cortical volume. And this is what we saw. So we use the contextual fear memory paradigm to form the contextual fear of one mice within one mice, and image the GFP signal when the mouse is receiving the context A training, and test another, when put the mouse in the other context and image the same signal, and we can see that the reporter show different activation in separate neuron ensembles. Although there are some neurons that show common activation. And within the same paradigm, if we compare two same contexts, then the neurons appear to be similar activated. So this allow us to compare different context-induced responses in the cortex. And this is what we really see from layer 1 through layer 5. And what you saw here is a 3D reconstruction of the confocal imaging on the right, and on the left, it's real imaging. And this imaging is composed of three pseudo-color. The red one indicates the reporter intensity in the home K trial. Green one compose the signal in the context A trial. And the blue one is the signal in the context C trial. And you can immediately see that in the layer 2, there are very great separations of 3 different contexts. While when the signals goes down in the layer 4, you see a lot of signals merged together. So that different layers may have different responses to formed memory. And for the black ones, it's a vessel which do not express EGR1. And the dots are actually the apical dendrites or collateral dendrites. So that we actually saw that signals in layer 2 show very great separation, while signal in layer 3 and layer 4 seems to be merged together. This makes us a little bit confused, because we know that the visual cortex is actually very specific for different stimuli. In different contexts, the context A and context C, the stimuli are changing so much why they activate the same population of neurons. And if we reconsider the context, so what context means is a temporal spatial combination of different components of the stimuli. And we think it's because the thing we saw here for the mixed signal actually reflects the temporal accumulation of different components induced response. For example, on time 1, the mouse is looking at the background, or on time 2 is concentrating on one door or some other place. So different part of the neurons might activated to respond specifically in the deep layers, while this makes different contexts seems to be similar due to the temporal accumulation effect. While if there is a neuron that continuously responds to the environment, to the specific context, no matter what the component it looks, then it will respond specifically to this context, formed invariant presentation. We want to evaluate this hypothesis. So if this is true, then if we use the signal in one context to minus the signal in another context, we might see very little changes in the layer 3, but we expect to see strong changes in layer 2. And this is what we saw. Most of neurons actually in this whole volume do not change much. And there consists about 3,000 neurons, and most of them form the signal changes formed a Gaussian distribution. While there's a heavy tail portion of the signals that show higher activation levels, and those neurons mainly localized in layer 2. And if we quantify all the cortical areas we measured, actually most cortical areas in response to different contexts show the similar distribution that only the layer 2, the superficial layer of the volume, showed very strong separation of the signals. While in the deep layers, the signal changes of the EGR1 GFP reporter did not show too much change. And this is consistent with the idea that the context-related memories may distribute mainly in superficial layers. And to further demonstrate this, we also use the simplified stimuli. So we anesthetize the mice and then use LED to generate simple stimulus on the visual system. And in this controlled experiment, we can see the activation of neurons throughout the cortical volume from layer 4 to layer 2. So this tells us actually the little changes in layer 3 and layer 4 in free-moving mice is due to the temporal accumulation effect. So the layer 2 neurons show the invariant presentation. Then we want to see whether this presentation is actually a memory-like response. We found the answer is yes, because those neurons, the response in those neurons actually are activated gradually through about 2 weeks, and the signal when the mouse experiences the same context increase gradually, and until about 15 days, and then it lasts a very long time until 1 month later, the response is still very high. While in the volume average, the signal in the cortical volume do not see so much changes. And also, we found that the signal in the layer 2 neurons also show stringent linear correlation with the recall of the contextual fear. The mouse freeze amount is linearly correlated with the signal. And also different contexts can find different population of neural ensembles, show very discrimination in these populations in response to different contexts. And also this memory-related signals can last for as long as 3 months. And the different cortical areas seems to have different dynamics for this context-specific response. The visual areas decayed a little bit, while the retrospinal area lasts for a very long time. And this signal or seeing in different cortical areas like motor cortex, visual cortex, and the somatosensory cortex. And it might imply that the memory traces is widely distributed across different cortical areas. So in a brief summary, using the EGR1 reporter, we saw the contextual fear memory-related cells mainly located in layer 2, while in layer 3 to layer 4, they may represent variable stimuli content. As the context is actually the temporal spatial combination of the layer of the different components. So it's very simple idea is whether this combination of the layer 3, 4 activities can contribute to the invariant presentation in layer 2. So to further ask this question, to demonstrate the question, we did further analysis. And a very short answer is maybe yes. We use the learning-induced signals to quantify the layer 4 neurons, and also we use these signals to generate a pattern which may mimic the synapse strength between the layer 2 and the layer 4 neurons. And if the formed memory connections is steady, then we can use this synaptic connection to predict the signals between layer 2 and the layer 4. And it should be a linear correlation between them. And yes, we found that this linear correlation. And before the training, we also saw a little bit linear correlation. However, the R-square is relatively lower. And also, you can also see that the slope is lower, means the strength between the layer 4 and the layer 2 neurons, specific layer 2 neurons, are a little bit lower than expected. So learning may strengthen this process. And this process, the strengthening during the memory consolidation, is hippocampal dependent. Because if we lesion the hippocampus, we found that this increase is totally abolished. And this is true for all the mice we have measured. We can also use our system to see the system consolidation in viral cortex. So in the system consolidation, the quick memory is formed in the hippocampus, and it will gradually remodel the cortical connection. So an implication is that you may see differential recall induced responses for recent and remote time. And whether we can see it, the answer is yes. In day 4, when we recall the contextual fear response, it's activate this neuron. However, 2 weeks later, if we look at the same memory recall task, it activates different population of neurons. And this steady presentation can last for 1 month, and we can see the same population is activated. And if we quantify them, we can see separated neuron populations. In a recent recall population, it responds fast and then decay. In a later phase, it lasts longer. And this separation is also hippocampal dependent, because when the hippocampus is a lesion, we saw the abolishment of this representation changes. The recall induced the steady presentation throughout 2 weeks. And it showed the significant more overlapping of the activating neuron. And this is not due to the sensation defect, because the activating neurons also different from the home cage ones. So to conclude our discovery, we found that context-specific neurons are formed in cortical layer 2, and the context-specific response is going through a process of, is formed through a process of consolidation. And also, hippocampus is required for both the correlation increase between deep layers and layer 2, and also it's required for the so-called system consolidation process. And this work's mainly done by Dr. Xie, who is also in audience here. And I thank and support from my from Li Hui that allow me to bring the EGR1 reporter to Tsinghua and quickly start our experiment. And thank you for your attention.
So I'm a little confused because in the mouse, the input from the geniculate comes into layer 4 and the bottom of layer 2, 3. So that's the input that is always going to be driven by different things,
Yes.
right?
Yes.
And I buy your idea that layer 2 cells may have already established through learning, a specific connectivity. But the output from the cortex is really from layer 4, layer 5 and 6. And one would expect, since they have to direct specific instructions to different parts, that they should have some specificity in terms of what they extract as well. And your data doesn't show anything down deeper.
In the deep layers of layer 5, I think layer 5 can also be divided to layer 5A and the layer 5B. And recent report have also shown that layer 5A might also receive thalamus input. However, in our study, the signal is kind of low because we haven't used higher power to trace those changes. And we mainly focus on layer 4 through layer 2 neurons. So in our study, we haven't touched layer 5 in a systematic way. So I couldn't answer your question now, but we would like to also look at that.
Quick questions.
Yeah.
I very like your computational model. Very nice color picture and the differentials staining. One quick question is that in your lesion study, you focus on the hippocampus formation areas, and in your model to start with is the contextual fear, and I believe amygdaloid should be involved. And also, I don't know much about the antiviral system in the mouse, but the smelling sense, the olfactory input is very strong. I wonder whether, in your next setting, try to lesion the, amygdaloid input and output, and also the, olfactory input to the neuron, and see how it might modify, the activity level at different areas.
Okay, thank you for your question. So for the fear conditioning, actually the QD fear conditioning is mainly involved in the amygdala. And contextual fear, hippocampus is also involved, and there's a lot of studies have been clearly showing that. And actually, we also found that if you lesion the hippocampus, the contextual fear response is for the long-term component is pretty much abolished. So we are sure that hippocampus is involved in this process. Secondly, the amygdala definitely, because it's a center for the fear, and its circuit have been shown to be involved in the memory formation. So we are also interested in how those amygdala circuits involved in this.
The last quick thing is that when you do the two-photon, imaging measurement, and one thing we might need to have a better control. For instance, the cerebral circulation blood flow to that particular regions. The memory brain, the neuron, they live along the watersheds and determined by the blood circulation distributions. And I believe the change of the cerebral blood flow at different region might have a partial contributions to the differential activity of the neurons at different layers that could serve a consideration for some confounding.
If that is the case, we might expect to see trial-to-trial differences because the changes in that. But actually, our system is quite consistent in this case report. So we did consider this as maybe factor changing our recording systems, but we measure that. Yeah.
Very good.
Thank you.
One last question there.
I might have missed this, but so you showed that the layer 2, when you put the mouse in a context and then change the context, there wasn't much overlap between the activated cells. I'm wondering if you just did the simple test of putting the mouse back in the same context and seeing the amount of overlap or reactivation.
Yes, I did show that. Yeah. So the context, in the same context, the same population of neurons could be activated. But if you look at the consolidation process, it's only after the system consolidation happened, they are very strictly in one population. But before that, it's still in a systematic evolution.